For this training, we will be isolating peripheral blood mononuclear cells or PBMCs from the whole blood using CPT tubes. To begin with, collect about 8 ml of peripheral venous blood in these tubes. Once the blood has been collected, these tubes must be stored upright and the blood must be processed within 2 hours of collection. To begin with, the blood is mixed by inverting the tube 8 to 10 times. This is done gently to avoid lysis of the red blood cells or the RBCs. Next, we centrifuge the blood for 30 minutes at 1500 RFC. The tubes are safely and securely placed in the buckets, making sure that they are well balanced. Upon centrifugation, the whole blood gets separated into different components. The top layer is the plasma, which is followed by the whitish layer of PBMCs and a clear layer of the density solution. The RBCs are found at the bottom below the polyester gel layer. At the end of centrifugation, check for hemolysis within the plasma layer. If the sample is hemolyzed, please annotate that in your sample collection form. The PBMCs are located in the whitish layer between the plasma and the density solution. If the thresholds between the layers is difficult to discern, collect both the white cell layer as well as the density solution for better yield. Before collecting the PBMCs, slowly aspirate about half of the unwanted plasma without disrupting the cell layer below it. Once we remove the excess plasma, collect the PBMCs below the plasma layer. Here I am trying to remove both the density solution as well as the PBMCs without disrupting the gel layer below it. Next, in order to wash the PBMCs, we are going to bring the volume up to 15 ml using 1x PBS solution. And then we'll gradually invert the tubes 5 to 6 times to mix this solution. Now the cells are ready to be centrifuged again. Next, we'll centrifuge our PBMCs for 15 minutes at 300 RCF at room temperature. At the end of the centrifugation, the cells would have pelleted at the bottom of the tube, as you can see here. Next, we remove the supernatant by slowly and carefully pouring the liquid into a waste tube without disturbing the cell pellet. The cell pellet then gets resuspended in 1 ml cryopreservation media that is provided by the Accurant Biotech. The cells are resuspended by up and down pipetting for 5 to 6 times. If the cells continue to clump, then repeat this motion of up and down pipetting, but do not do that more than 20 times, otherwise it will lead to cell lysis. The resuspended cells are then aliquoted into 2 tubes. Aliquot 800 microliters in 1 cryotube for ADCC assay and transfer the remaining volume to a second cryotube for NK phenotyping. Each subject at each visit will generate 2 cryopreservation tubes for each of ADCC and NK assay, one as primary and the other as backup. Store the cryopreservation tubes in Corning cool cell freezing containers at minus 80 degrees Celsius freezers for overnight cooling. The following day, these tubes can be removed from the freezer and stored in nitrogen tank, which is highly recommended. Before putting away the vials into the minus 80 freezer, here are the few things to keep in mind. The Corning freezing container controls the rate of freezing by minus 1 degree Celsius per minute when stored in a minus 80 freezer. Here, ensure that the box and the vials are kept in an upright position. If the freezing batch is fewer than 12 vials, fill each well with a cryopreservation vial that contains the equivalent volume of freezing media. And the last thing to remember, don't ship primary as well as the backup vials of the same visit from the same subject in the same shipment.